Manjunath here. Okay, let us start with the next part of the chapter, that is principles of inheritance and variations. So, in the previous part of the video, we have discussed about the monohybrid cross, dihybrid cross, and particularly about the chromosomal theory of inheritance we have finished. Right. Coming to the next part of the chapter, that is deviation from Mendelian genetics. Okay, deviations from Mendelian genetics. Okay, what do you mean by deviations from Mendelian genetics? First of all, see, Mendel have given some of the propositions. Okay, he has given some of the laws, and he told that, okay, all of these rules or all of his rules or laws are applicable universally. Okay, they were applicable universally, but okay, what happened when the scientists have carried out the Mendelian experiments on different plants and different organisms, they did not get the same results as that of Mendelian results, and that is how they told that the Mendelian laws are applicable not for all the individuals or all the organisms, but they are applicable to only certain limited organisms. Okay, so Mendel told that his laws are universal, but the thing is, his laws are not universal because he did not test the same laws in what? Other organisms, right? And so those deviations are found, okay, in that first one is codominance. Okay, in that first one is codominance. What is codominance? Okay, see, it is a condition in which both genes are expressed for a particular character in the F1 generation. Okay, it is a condition in which both genes are expressed. Okay, in case of Mendelian law of dominant, okay, law of dominance, what he had told, he told that in the F1 generation, okay, the gene which is going to express is called as dominant gene, and the other gene which is not going to express is called as recessive gene. And what he had told, he told that in the F1 generation, okay, only one gene will get expressed, which is called as the dominant gene, but in that particular pea plant it was applicable, but in few organisms, in some other organisms, if you take for example, that particular rule is not applicable. Okay, and by means of codominance we can prove that, okay, the Mendelian laws are not applicable for all the condition. Why? Because in some of the organisms we can see that, okay, the condition in which both the genes are expressed or both the genes are dominant in nature, okay, and there is no blending, even though they are dominant, there is no blending. Without blending they will get expressed, okay, they will get expressed equally. Is that clear? What is codominance? See, I will repeat, codominance is a condition in which both genes will get expressed for a particular character and they will get expressed equally without any blending, without any mixing, they will get expressed equally. Okay, and the examples for codominance, okay, are what? See, the common examples for codominance are AB blood group in human beings, okay, AB blood group in human beings, and the carrier sickle cell anemia, okay, the carrier of sickle cell anemia. See, in case of AB blood group in human beings, okay, you already know, or else if you don't know, just listen here. In case of human beings, okay, the blood group is controlled by, the blood group is controlled by a particular gene I, and this gene I has got two dominant alleles, they are IA and IB, and only one recessive allele is there, that is small I. Okay, Of these, when the individual has got IA and IA, it will lead to the blood group A. And blood group A is also resulted by presence of one dominant gene, that is IA, and one recessive gene, that is small I. Similarly, the B blood group can be found, okay, by means of having two dominating genes, that is IB, or else one dominating gene, that is IB, and one recessive gene, that is small I. But when you go to the blood group AB, it is resulted when both the dominating gene occur at a time, that means IA and IB, okay. Both dominating gene occurs at a time, that is IA and IB. When IA and IB are dominating genes, when they occur together, it will lead to the formation of what? AB blood group. Okay, it will lead to the formation of AB blood group. It means here antigen A as well as antigen B both are produced. Okay, that means expression of antigen A as well as antigen B, or expression of gene IA and gene IB, both have been taken place there. Okay, both are expressed equally. Okay, both are expressed equally. So that is called as what? Co-dominance. Okay, both the alleles for a particular trait will be what? Dominant. Okay, both the genes will be dominant, right? So they will get expressed equally. That condition is called as co-dominance. Okay, and even you can observe this particular co-dominance in the particular disease, that is sickle cell anemia. Okay, an individual who is carrier for sickle cell anemia will show what? The condition HbA and HbS, where both are dominant here. Okay, where both are dominant, and this dominant individual or this carrying individual will have what? The co-dominance condition, right, which is an exception or which is a deviation from Mendelian genetics, because Mendel told that either of them will be dominant. Okay, one will be dominant and one will be recessive, but here both are dominant. Okay, both are dominant. That is how the deviations are seen from Mendelian genetics, and the first one is co-dominance. Coming to the next one, that is pleiotropic gene. Okay, next one is pleiotropic gene. It is another deviation. Okay, what is pleiotropic gene? See, it is a gene which controls more than one character. Okay, it is a gene which controls more than one character. According to Mendel, what has happened? One gene, okay, one gene controls one character. Yes or no? According to Mendelian genetics, one gene control one character, but the deviation from Mendelian genetics shows that pleiotropic gene is also existing. And what is pleiotropic gene? It is a gene which controls, okay, more than one character. Okay, single gene, if it controls more than one character, then it is called as what? Pleiotropic gene. Okay, and the best example is what? Sickle cell anemia. Okay, sickle cell anemia. In case of sickle cell anemia, single particular gene controls more than one character. Okay, next one, even you can give the example of phenylketonuria. Okay, phenylketonuria can also be formed. Next example for the deviation from Mendelian genetics is multiple allelism. Okay, Mendel told that for a particular character there will be two alleles, two varieties. Okay, only two varieties, or else two alternative forms will be there. Say for example, when he has taken the tallness, okay, or else height of the plant, okay, when he has taken the height of the plant as an example, it had got tall and dwarf varieties only. Only two varieties were there, because the genes were only two, or else alleles were only two, capital T and small t. Okay, for the character, that is height of a plant or height of the stem in case of pea plant, have got only two varieties, that is capital T and small t. But when we have observed the same thing in other organisms, we found that a gene may have, a gene or a character may have several varieties due to presence of multiple alleles. A particular gene may have what? Multiple alleles or more than two alleles. A gene may have more than two alleles. Say for example, in case of human blood group ABO. Human blood group ABO. In case of humans, the blood group is controlled by the gene I, as I have already told you in the previous slide. The blood group is controlled by gene I. This gene I has got what? Three alleles. That is, first allele is capital IA, second allele is capital IB, and third allele is small I. There are three alleles for a particular character, or for a particular trait in the blood group, human blood group. So here, see, more than two alleles are occurring, that is why it is called as multiple alleles. The condition is called as multiple allele. What is multiple allele condition? It's a condition in which more than two alternative forms of the same gene occur. That is called as multiple allele, right? And they are formed due to mutation and located on the same locus of homologous chromosome. This may be asked for your NEET examination. The multiple alleles will be present or will be located on dash. They will be located on same locus of homologous chromosome. They will be located on what? Same locus of homologous chromosome, right? And some important points I have given here for your information, right? See, the first one is Dr. Karl Landsteiner, got Nobel Prize in 1930 for the discovery of blood groups A, B and O. He have discovered the blood group A, B and O, but the blood group AB was discovered by another person called as D. Castello and Sturley. D. Castello and Sturley have discovered AB blood group. Karl Landsteiner, got Nobel Prize for the same thing, right? Very important point these two are. Very important point. Who have got Nobel Prize for the discovery of AB and O blood group, AB and O blood group, that is Karl Landsteiner. And who have discovered AB blood group? It is D. Castello and Sturley. Next point, blood groups such as M, N, Mn were discovered by Landsteiner again, as well as A.S. Wiener. Landsteiner and Wiener together discovered some other types of blood group, rare type of blood groups called as M, N and Mn. Is that clear? Next one, Rh factor E1. The Rh factor E1 was discovered by Karl Landsteiner and A.S. Wiener. They have also discovered the Rh factor. That means the Landsteiner, he started to work on the particular process or particular blood grouping continuously. And ultimately what he had done? He had achieved many of the things, that is, he have discovered the blood groups A, B, O, as well as he had discovered the blood groups M, N and Mn, and he also discovered what? Rh factor. That is why he is the most important person when you start talking about what? The blood group. You should remember that this particular person, because in the NEET examination the question may be asked, who have discovered Rh factor or who have discovered the blood grouping, right? Next one, lethal genes. Lethal genes. The next one is lethal gene. Another example. What is lethal gene? What is lethal gene? Do remember, it is the gene which causes death. The gene that causes death of the individual in early stage when it comes in homozygous condition. What is lethal gene? Any gene if it occurs in homozygous condition, okay, any gene if it occurs in homozygous condition due to which it causes the death of the individual is called lethal gene. ಅಂದರೆ ಯಾವುದಾದ್ರೂ ಒಂದು ಜೀನ್ ಹೋಮೋಸೈಗಸ್ ಕಂಡೀಷನ್ ಒಳಗಡೆ ಬಂದಾಗ ಇಫ್ ದ ಇಂಡಿವಿಜುವಲ್ ಡೈ ಇನ್ ದ ಅರ್ಲಿ ಸ್ಟೇಜ್ ದೆನ್ ಇಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಲೀಥಲ್ ಜೀನ್. ಓಕೆ, ಒಂದು ಯಾವುದಾದ್ರೂ ಜೀನ್ ಹೋಮೋಸೈಗಸ್ ಕಂಡೀಷನಲ್ಲಿ ಬಂದಾಗ, ಓಕೆ, ಇಫ್ ಇಟ್ ಕಾಸಸ್ ದ ಡೆತ್ ಆಫ್ ದ ಇಂಡಿವಿಜುವಲ್ ದೆನ್ ಇಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಲೀಥಲ್ ಜೀನ್. ಓಕೆ, ಆ್ಯಂಡ್ ಇಟ್ ಮೇ ಬಿ ಡಾಮಿನೆಂಟ್ ಆರ್ ರಿಸೆಸಿವ್, ಬಟ್ ಮೋಸ್ಟ್ ಆಫ್ ದ ಟೈಮ್ಸ್ ಇಟ್ ವಿಲ್ ಬಿ ರಿಸೆಸಿವ್ ಫಾರ್ ಲಿಥಾಲಿಟಿ. ಓಕೆ, ಡಾಮಿನೆಂಟ್ ಆದರೂ ಇರ್ಬೋದು, ರಿಸೆಸಿವ್ ಆದರೂ ಇರ್ಬೋದು, ಆದರೆ ಮ್ಯಾಕ್ಸಿಮಮ್ ಟೈಮ್ಸ್ ಇಟ್ ವಿಲ್ ಬಿ ಮೋಸ್ಟ್ಲಿ ವಾಟ್ ರಿಸೆಸಿವ್ ಫಾರ್ ಲಿಥಾಲಿಟಿ. ಆ್ಯಂಡ್ ದಿಸ್ lethal gene was discovered by, okay, the Q0 in the coat color of the mice. Okay, right, LQ0 in the coat color of the mice. And in case of humans, you have, or you can see this particular example of lethal gene. Okay, sickle cell anemia or HBS is an example for what? Lethal gene. When the HBS occurs, okay, when the HBS occurs in homozygous condition, it will lead to, okay, when the HBS occurs in homozygous condition, it will lead to what? The sickle cell anemia, and it will lead to death of the individual. Is that clear? Coming to the next one, that is epistasis. What is epistasis? Okay, see, a gene which prevents expression of another gene or non-allelic gene, then it is known as epistatic gene. Usually what happens, see, a dominant gene will prevent the expression of a recessive gene. If you consider capital T as a dominant gene, okay, dominant gene, it always prevents the expression of, or it always stop the expression of what? The recessive gene. This is in normal condition. Normally it will happen. Normally dominant gene will prevent the expression of recessive gene. Okay, how these are controlled here? Both of these are what? They are located on the homologous region or homologous condition. They will be present on the homologous, or else they will be present as the alleles of a same gene or same character. They will be present as alleles for a same character or same gene. ಅಂದರೆ ಕಾಮನ್ ಆಗಿ ನಮಗೇನು ಗೊತ್ತಿದೆ ಅಂತಂದರೆ, ಡಾಮಿನೆಂಟ್ ಜೀನ್ ಏನು ಮಾಡುತ್ತೆ, ರಿಸೆಸಿವ್ ಜೀನನ್ನು ಸಪ್ರೆಸ್ ಮಾಡುತ್ತೆ. ಇಟ್ ಈಸ್ ಇನ್ ಕಾಮನ್. ಬಟ್ ವಾಟ್ ಈಸ್ ಎಪಿಸ್ಟಾಸಿಸ್ ಅಂತಂದರೆ, ಇಟ್ ಈಸ್ ಅ ಕಂಡೀಷನ್ ಇನ್ ವಿಚ್ a gene, okay, a gene prevents the expression of another non-allelic gene, okay, another non-allelic gene. And to remember, the gene which prevents the expression of non-allelic gene, it may be dominant or it may be recessive. Okay, it may be dominant or it may be recessive, but it is going to control the expression of what? Non-allelic gene. Okay, it is going to control the expression of non-allelic gene. Non-allelic ಅಂತಂದ್ರೆ these are alleles, capital T small t ಅಂದ್ರೆ alleles, ಒಂದೇ ಒಂದು ಕ್ಯಾರೆಕ್ಟರ್ಗೆ ರೆಸ್ಪಾನ್ಸಿಬಲ್ ಇದ್ದಂಥದ್ದು. Okay, these are responsible for the production of only one character, that is height of the plant. Okay, height of the plant ಅನ್ನು ಇದು ಡಿಸೈಡ್ ಮಾಡುತ್ತೆ. ಆದರೆ ನಾನ್ ಅಲಿಲಿಕ್ ಅಂತಂದ್ರೆ it is totally from different part of the what? Gene. Okay, idu allelic antandre which are related to the same character or which are present in the same region, but non allelic andre totally different gene will be showing its influence on other gene, so that is called as non allelic. Is that clear? Next one, the expression of gene which is suppressed by the epistatic gene. Okay, ಯಾವ ಜೀನ್ ದ ಎಕ್ಸ್ಪ್ರೆಷನ್ ಸಪ್ರೆಸ್ ಆಗಿರುತ್ತೆ ಅದನ್ನೇನಂತ ಕರಿತೀವಿ? ಹೈಪೋಸ್ಟಾಟಿಕ್ ಜೀನ್ ಅಂತ ಕರಿತೀವಿ. ಇಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಹೈಪೋಸ್ಟಾಟಿಕ್ gene. The expression of the gene which is controlled by the epistatic gene, it is called as what? Hypostatic gene. Okay, complementary genes. Okay, the next part is what? Complementary genes. So what exactly is meant by complementary gene? it is an another example for what? Okay, it is another example for deviation from Mendelian genetics. Okay, it is another example for what? Deviation from Mendelian genetics. So, coming to the complementary genes. So, here say two pairs of non-allelic genes are essential in dominant form to produce a character. Okay, for a particular character to be produced, what is required here? Two pairs of non-allelic genes. Okay, two pairs of what? Non-allelic genes are required in dominant form. Okay, two pairs of what? Non-allelic genes are required in dominant form to produce a particular character, and that is called complementary gene. Is that clear? And such genes act together to produce an effect. Neither can produce its effect separately are called as complementary gene. It means the genes or complementary genes are those which will be producing one particular character only when they occur in pair, that too in the dominant condition. But if they are occurring in single, that means in unpaired condition, even though they are dominant, they will not give rise to any of the character. Okay, they cannot produce a particular character. Say for example, if a character A is produced by the complementary genes B and C in their what? Dominant condition. Okay, so if the condition is B and C, then only the character will be produced. Okay, if the condition is B and C, then only what will be produced? The character will be produced. In case if it is only capital B, or if it is only capital C, then this cannot be produced. Is that clear? What is a complementary gene? It is two pairs of non-allelic genes. Two pairs of what? Non-allelic genes. That means dissimilar genes. Okay, should be occurring in what? Dominant form. Okay, they should occur in dominant form, then only they can produce one particular character. Okay, this type of genes are called as complementary genes. Okay, the example of complementary genes can be seen in what? The Lathyrus odoratus plant. Okay, the flower color in case of the Lathyrus odoratus plant, you can find the complementarity, or else the complementary genes are actually present with this particular character of the plant, that is Lathyrus odoratus. Okay, Lathyrus odoratus. Sorry. Next one. Coming to the next one, that is supplementary gene. So, what is meant by supplementary gene, which is also called as recessive epistasis? Okay, a pair of gene. A pair of gene change the effect of another non-allelic gene, that is called as supplementary gene. Okay, a pair of gene changes the effect of another non-allelic gene. If a pair of gene changes the effect of non-allelic gene, then it is called as supplementary gene. Okay, which is also called as recessive epistasis. Okay, but dominant epistasis, what would have happened there? In case of dominant epistasis, they used to mask the expression of one particular character of a gene. Okay, non-allelic gene's character used to be masked. That is why epistasis is generally known as masking of the genes. Right. And here supplementary gene is what? It is an example for recessive epistasis. Right, where a pair of gene changes the effect of another non-allelic gene. Okay, a pair of gene changes what? Effect of non-allelic gene, that is called as supplementary gene. And the example is coat color in mice. Okay, and the example is coat color in the mice. So, next one, let us go to the next one, that is incomplete dominance. It is the best example to explain what? The deviation from Mendelian genetics. Okay, incomplete dominance. What is the first law of Mendel's proposition, or what is the first law proposed by the Mendel? The law of dominance. Is that clear? The law of dominance. According to law of dominance, what it suggests? Okay, according to law of dominance, what it suggests? In the heterozygous condition, okay, in the heterozygous condition, okay, say for example capital T and small t, okay, one of the character will be produced and that character, okay, is considered as what? Dominant, and the gene responsible for that character is considered as dominant gene. But the incomplete dominancy is an exception, okay, where, see, incomplete dominancy is an exception where what happens? The dominant gene, okay, and recessive gene, okay, both of them will be not completely expressed or not completely left. That means here what happens in the incomplete dominancy, it is a condition in which, see, it is a condition, incomplete dominance, it's a condition in which dominant gene, okay, dominant gene fails to suppress the recessive gene or dominant allele, okay, you can call it as dominant, okay, allele fails to suppress recessive allele in heterozygous condition, okay, in heterozygous condition. Is that clear? As a result, what will happen? As a result, okay, as a result, an intermediate character is obtained. Okay, as a result, what will happen? An intermediate character will be obtained. So that is called as what? The incomplete dominance. That means the dominance will be incomplete. Okay, the dominant gene will be incompletely dominant over a recessive gene. See here, coming to the first point. According to Mendel's law of dominance, dominant character must be present in F1 generation. But in some organisms, F1 generation is different from both the parents. Okay, F1 generation, okay, will be different from what? Both the parents. Why it will be different from both the parent? Because what will happen? The dominant gene fails to suppress the recessive gene completely. Okay, dominant gene fails to suppress the recessive gene completely. As a result, both the genes will get, or both the alleles will get expressed. Due to expression of both the alleles, what will happen? An intermediate character will be seen. Okay, so that itself is explained further. See here, incomplete dominance was first discovered by Correns. Important point here. Okay, do remember, incomplete dominance was first discovered by Correns in the plant called as Mirabilis jalapa. In the plant Mirabilis jalapa, what has happened? Incomplete dominance was first discovered. Okay, and this Mirabilis jalapa is commonly known as 4 o'clock plant. Okay, it is commonly known as 4 o'clock plant, or else the gul beans. Okay, next one. Okay, this particular 4 o'clock plant, in Kannada it is known as Sanjay Mallige. Okay, in Kannada it is known as Sanjay Mallige. You can call it as, okay, in Kannada you can call it as Sanjay Mallige. Evening 4 o'clock plant, at evening 4 o'clock the flower will blossom. That is why it is called as 4 o'clock plant or Sanjay Mallige. Right. Next point, this particular plant shows what? The incomplete dominance. See here, 3 different types of plants are found in Mirabilis, okay, on the basis of flower color, such as red color, white color and pink color. That means Mirabilis jalapa or this 4 o'clock plant have got 3 different types of flowers regarding to their color. Okay, with respect to their color, there are 3 different types of flowers produced in this particular plant. Okay, when the plants with red flower is crossed with the plant with white flower, then what will be obtaining? Pink flower is obtained. Remember, when the plants, okay, when the Mirabilis jalapa plants having red colored flowers, okay, having red colored flowers are crossed with another Mirabilis plant having white colored flower, okay, another Mirabilis plant having white colored flower, then the resulted plant have got pink colored flower. Okay, the resulted plant have got what? Pink colored flower. The reason of this is that the genes of red color is incompletely dominant over, okay, the genes of red color is incompletely dominance over genes of what? White colored plant. Okay, the gene of red colored plant is incompletely dominant over the genes of white color. Okay, that I will explain you with the help of what? The cross now. See, when, okay, when F1 generation of pink flower is selfed, then in the F2 generation, okay, the gene ratio that is obtained is red, pink and white will be what? 1:2:1. Okay, in place of normal 3:1 ratio of Mendel. Okay, and the same I would like to explain in this particular part here. See, when the parents of, or when the red colored, okay, when the red colored plant is crossed with white colored flowering plant, okay, color of the flower is red in one particular plant and it is crossed with another plant having white color. So, this is phenotype of the parent, okay, phenotype of the parent. Then the genotype, okay, genotype is what? Capital R and capital R crossed with small r and small r, right. Then what about the gametes? The gametes will be capital R crossed with small r. Okay, it is resulted by means of meiosis. Okay, meiosis, right. Next one, when both of these are united to form what? Capital R and small r. Here what has obtained? Okay, F1 generation is been obtained, F1 generation, and all the individuals which are produced in F1 generation, okay, according to Mendel, they have to get what? The character of any one of the parent, because according to dominancy of law of Mendel, according to Mendel's law of dominancy, in F1 generation the offspring should get any one character from the parent. That means either it should show red colored flowering plant or it should show white colored flowering plants. But surprisingly, in this particular plant we have got, okay, or Correns have got what? The plant which were having pink colored flowers, okay, pink colored flowers. Then he got confused and he crossed what? F1 into another F1. Then what has happened? Capital R small r is crossed with capital R small r, right. Then this is what? The phenotype of the parent. Then the, sorry, genotype of the parent. And what about the gametes? Okay, the gametes is capital R and small r crossed with capital R and small r, right. So, when this one is done, then let us put the Punnett square. Okay. Okay, this is male and here it is female. The male individual has got capital R, small r as their gametes, female have got capital R and small r as their gamete. So, in the first one capital R and capital R, here capital R and small r, here capital R and small r, here small r and small r, right. Due to presence of both the genes or both the alleles as capital R, here red colored flowering plants are produced, and here due to presence of one capital R, one small r, in both of these pink colored flowering plants are produced, and here also pink colored flowering plants are produced, and in the second generation, in the last one, that is the white colored flowering plants are produced. Okay, white colored flowering plants are produced. So by means of this result, okay, see here, red colored flowering plant is only one, okay, and pink colored flowering plant, okay, it is 2 in number, and white colored flowering plants, it is 1 again. It means, okay, it means the phenotypic ratio of this incomplete dominance is, the phenotypic ratio of incomplete dominance is 1:2:1. At the same time, what about the genotypic ratio? Capital R is only 1, okay, then capital R and small r, they are 2 in number, then small r and small r, that is only 1 in number. So phenotypic ratio as well as genotypic ratio, both are same, okay, phenotypic ratio and genotypic ratio both are same here, clear. So here in this particular representation, you can easily understand, see where exactly the deviation has been taken place, okay, where you can find the deviation exactly. See, first deviation here in this particular F1 generation, okay, normal individual, okay, according to Mendel, okay, what should be the result? The result should be what? Either red or white, but here the result is intermediate, that means pink. Neither red colored flowering plants is obtained nor white colored flowering plant is obtained. Instead, what has obtained? The pink colored flowering plant has been obtained. Why this is pink colored? Because here what has happened, the R, which is called as dominant allele, is not suppressed the recessive allele completely, okay, has not suppressed the recessive allele completely. That is why both the characters are expressed here, both the characters are expressed. That is why what has happened, the flower color have got turned into pink, okay, both pink and both red and white color, if they get mixed, then it will give rise to pink coloration. Like the same, they have obtained the pink colored flowering plant in the first generation, okay. And when, see, look at this particular point, first point of this slide, when F1 generation of pink flower is self pollinated, then phenotypic ratio of F2 generation is red, pink, white, that will be equal to 1:2:1. Yes or no? See, we have done it already. When F1 generation individuals are self crossed or self pollinated, then what has happened? In the F2 generation, this is F2 generation. Yes or no? In F2 generation, okay, the ratio of red, pink and white is 1:2:1. Okay, and the actual monohybrid cross of Mendel have got what? 3:1 as the, okay, 3:1 as the monohybrid cross phenotypic ratio. But here, even though it is monohybrid, the phenotypic ratio is varying, okay, that is 1:2:1. Why the phenotypic ratio is varying like this? Because it is showing incomplete dominance. Okay, if it is completely dominant, then it would have given the result of 3:1. Since it is incompletely dominant, it has given the result of 1:2:1. Is that clear? So that is the phenotypic ratio, and even the genotypic ratio is 1:2:1. And very important point to be noted here is, okay, very important point to be noted here is, even though it has produced pink color here, even though it has produced pink color here, in the F2 generation, again we have got red colored flowering plants as well as white colored flowering plants. Okay, even though all the F1 individuals are pink in nature, but when the F1 individuals are self crossed or self pollinated, in the F2 generation we have got red as well as white color, which are of what? The parental characters. Okay. It means that, okay, it means or it explains very clearly that the characters will not undergo any mixing or blending. Okay. Genes will never undergo blending. Okay. They will be present in their purest form always. Okay. I think you have already studied about what? Law of purity of gametes. Law of purity of gametes. Like the same here, the law of purity of gametes is again maintained properly. That is why ultimately in the second generation we have got what? The parental forms. If the genes or if the characters or traits, if they have blended or if they have mixed up with one another to produce the pink coloration, then in the second generation, okay, we could not have got the red and white colored flowering plants. Andre even though illi pink colored flowering plant bandide ಆದರೆ ಏನಾಗಿಲ್ಲ ಅಂದರೆ the genes have not combined with each other. ಎರಡು ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಮತ್ತು ಸ್ಮಾಲ್ ಆರ್ ಎರಡು ಕಂಬೈನ್ ಆಗಿಲ್ಲ. they are in their pure form only. ಅದು ಹೆಂಗೆ ಗೊತ್ತಾಯಿತು? when we have crossed, okay, when we have made the self cross of F1 individuals, then we came to know that, okay, the genes are not combined with each other. There is no blending. Okay, blending of the genes will not take place ಅನ್ನೋದು ಕ್ಲಿಯರ್ ಆಗಿ ಗೊತ್ತಾಗ್ತದೆ. Is that clear? So this is what incomplete dominance. And last point, look at this last point. Incomplete dominance is also seen in the plant called as Arenitum. Okay, okay, Antirrhinum, sorry, it is also present in Antirrhinum majus plant. Okay, it is also present in Antirrhinum majus plant, and this plant is also known as a snapdragon or dog flower. Okay, Antirrhinum, it is also known as snapdragon or else dog flower plant. Clear? This is what incomplete dominance. And do remember, for the theory it can be asked for 5 marks, but since you are preparing for the NEET examination or CET examination, you have to remember not the complete part but the part which is very very important, that is phenotypic ratio and genotypic ratio. In the CET examination you may get the question, name the cross, okay, name the cross in which the phenotypic ratio and genotypic ratio in F2 generation is same. Name the cross in which the phenotypic and genotypic ratio in the F2 generation is what? Same. That is phenotypic ratio 1:2:1, genotypic ratio 1:2:1. So that is what? The incomplete dominance. Right? Let us go to the next one. Okay. The next one is linkage. Okay. The next one is linkage. What is linkage? Okay. By the common English terminology, linkage you can understand. Okay. Linkage is the connection between any two things. Okay. It may be any gene or it may be any two things. Okay. And here, since we are dealing with the genes and the DNA, here the linkage refers to what? Staying together of certain genes during the inheritance. Okay, during the inheritance, that means during the transfer of characters from one generation to other generation, what happens? Segregation should take place. Okay, but sometimes what happens? Without proper segregation, some two characters which are maintained by two genes will be occurring together. Okay, they will not show any segregation. Okay, they will not show any segregation. Okay, that itself is called as linkage. But in general we can say that linkage is physical association between, see I will write it here, physical, okay, physical association, physical association between any two genes on a particular chromosome. Physical association between any two genes on a chromosome is called as what? The linkage, physical association between any two genes on a chromosome is called as linkage. Right. Next one. Linkage was first seen by Bateson and Punnett. Okay. William Bateson and Punnett for the first time have observed the linkage in the particular plant called as the Lathyrus odoratus. Okay. In the particular plant Lathyrus odoratus, they have observed the linkage and gave coupling and repulsion phenomenon. They have explained what? Coupling and repulsion phenomenon. Coupling means getting together, repulsion means to reject or else to get separated. Right. Those phenomenon were given by the Punnett and Bateson, but they did not explain the phenomenon of linkage. Okay. They did not explain the phenomenon of linkage. What they have done? They have just observed the linkage for the first time, but they were unaware of the linkage phenomenon properly. Okay. The proper idea of linkage was not there for Bateson and Punnett. Even though they have observed it for the first time, they were completely unaware of that, and that is why they have given 2 points, that is coupling and repulsion, but they did not explain that is the phenomenon of linkage. Okay. But the phenomenon of linkage was first explained properly by the T.H. Morgan. Okay. See, sex linkage was first discovered. Sex linkage was first discovered by T.H. Morgan. Okay. And you know that T.H. Morgan is what? He is the father of experimental genetics. T.H. Morgan is the father of experimental genetics, and this T.H. Morgan have first discovered what? Sex linkage. Okay. That means linkage between any two genes on the sex chromosomes. Okay. Linkage between any two genes on the sex chromosomes, that is called as sex linkage, and the sex linkage was observed by Morgan in the particular insect, that is Drosophila melanogaster, commonly called as fruit fly. Okay. In the Drosophila melanogaster, he has observed the sex linkage and he has proposed the theory of linkage there. Okay. By means of observing, he has proposed what? Theory of linkage. Is that clear? The next one, linkage and independent assortment can be represented in dihybrid plant. Okay. Linkage and independent assortment can be represented in dihybrid plant, because if it is a single gene, then linkage cannot be explained. Okay. Because linkage will be present between any two genes of any two different characters. Okay. Two different genes of two different characters. But in case of monohybrid, or in case of the example where you take only one trait or only one character, then linkage cannot be expressed. Right. To express the linkage or to explain in detail about the linkage, you have to take the example of what? A dihybrid cross itself. Okay. That is why linkage and independent assortment can be expressed or can be represented in the dihybrid plant. Then next one, linked genes are linearly located on the same chromosome. Okay. Linked genes, right? I will draw it here. Okay. You just observe. I will draw the structure, how they will be. If you consider this as a chromosome, linked genes will be present on the same chromosome linearly. Okay. That means they will be present on the same chromosome, okay, linearly. That means one after the other. Right. Next one, they get separated if exchange or crossing over takes place. Okay. They get separated if exchange or crossing over. See, I draw the chromosome structure here. If you consider this as one chromosome and this as another chromosome, homologous chromosomes. Okay. During the crossing over, what will happen? Okay. During the crossing over, I will draw them. They show the crossing over like this. Okay. But where the genes will be, as I have shown, they will be present one near to the other or linearly. It means if the gene one is there here, the second one will be there here. Okay. During crossing over, what will happen? They will get exchanged, okay, or else they get separated or exchanged in the crossing over. Is that clear? Right? I will show you with the diagram there. Okay. Before going to types of linkage, I will show you how exactly the linkage will be seen. See, if you consider this as one particular chromosome, okay, and this as another chromosome, homologous chromosome. So, if the gene A is there here and gene B is there here, okay, this is gene A and this is gene B, or if it is the allele or two genes, okay, A and B are there here. After crossing over, what will happen? After crossing over, see, after crossing over, like this, they will get crossed over, and I will just indicate with the different color, okay, the same homologous chromosome, but with the different color I will indicate. Hope you can clearly see this one, okay. Right. So, they have undergone crossing over. After crossing over, what will be produced? See, no doubt these two arms of the chromosome are have not undergone any crossing over. That is why they will be as it is. As a result, sorry, wait, I will change the proper coloration. I will give the proper coloration like this. See, the first chromosome will remain as it is, the first what arms of the chromosome, first chromosome, they will remain as it is, but when you go to the second arms, they half will be, okay, half of them will be what, normal, but the other half, okay, but the other half will be exchanged, but the other half will be exchanged with what, the another one, like this. At this time, what has happened? The gene B, okay, the gene A will be present at the same time, but the gene B will be present on, see here, right. The gene B will be transferred to the another one. By the end, what will be happening? This type of chromosomes will be, okay, resulted. Okay, at that time, what has happened? The gene A will be there as it is here, but gene B which is present here, it will be shifted to the other chromosome. Is that clear? That is why they get separated. The genes will get separated. Okay, the genes will get separated during the crossing over. Is that clear? The genes will get separated during crossing over, like this. Is that clear? After crossing over, exchange of the arms of the chromosome will take place. Due to that, what will happen? The exchange of or separation of the genes will take place. Okay, separation of the genes will be seen. After crossing over, separation of the genes will be seen like this. Clear? This will lead to what? The formation of non-parental combination. Okay, non-parental combinations will be resulted by means of the crossing over. Okay. Next, see the crossing over, okay, the linkage. Linkage, if it is very strong, that means if the genes are placed very near to each other, say for example, here in case of the diagram which I have drawn, okay, the gene A is there here and gene B is there here, which are very far away from each other, yes or no? If they are present very near to each other like this, if you consider this as gene B, okay, and this as gene A, then what will happen? Separation will not take place even after crossing over. Is that clear? That means if the distance, okay, if the distance between the genes, if the distance between genes is more, then what will happen? Recombination will be more. Recombination means their separation possibility, okay, possibility of getting separated from each other will be more. Okay, but if the distance between the genes is less, then recombination will be less. That means the possibility of getting separation from each other will be less, right. Let us move to the next one, that is types of linkage. Okay, that is types of linkage. See, in linkage you can find different types. Okay, in linkage you can find different types, such as the first one, see here, complete linkage. Complete linkage is a type of linkage in which the genes are always show the parental combinations itself. Okay, what do you mean by parental combination? Okay, that I will show you here. See, if you consider these as the chromosomes, okay, on which, okay, these are the chromosomes on which the two genes are located. Okay, that is gene A here and gene B. Then what will happen? If the genes are located in very near position to each other, it means that the linkage is very strong, because even after crossing over they cannot get separated. Okay, even after crossing over they cannot get separated. So what are the parent here? One have got A and B gene and other have got no gene. So even after getting crossing over, what will be there? The chromosomes will be present as it is without any change, yes or no, because the genes are present very near to each other. The chromosome will remain as it is. One will be having A and B and other will be having nothing in it, right. So this is what the parental combination. Okay, parental combination means what? See, here one chromosome has got A and B, both the genes, that is A and B, and 0 here. Okay, so this is called complete linkage. In case of complete linkage, even after, okay, in case of complete linkage, even after crossing over, the parental combination will not be lost. Okay, parental combination will not be lost. So such type of crossing over or such type of linkage is called as what? Okay, such type of linkage is called as complete linkage. Okay, what is complete linkage? It is a type of linkage in which, okay, the genes always show parental combination because they are linked very tightly. Why they are said to be linked very tightly? Because they cannot be separated. Why they cannot be separated? Because they are present very near to each other. That means distance between those two genes on a chromosome are very small. Okay, the distance between the genes on a chromosome is very small or very less, and that type of linkage is called as complete linkage. Okay, that type of linkage is called as what? Complete linkage, and they will never form new recombinations. Okay, they can never form new recombination, only parental combinations can be produced. Okay, no recombinations can be produced here. But and next point, crossing over is absent in it. Okay, such genes are located very close on the chromosomes, and such type of linkage are very rare in nature. Okay, example, male Drosophila, okay, female silk moth. These two are the example which show, okay, which show no recombination. You cannot find any of the recombination in case of male Drosophila. Okay, the male Drosophila will not show any linkage. Okay, the male Drosophila will not show any linkage, right, as well as the female silk moth. Coming to the next one, incomplete what? Linkage. Incomplete linkage, it is a type of linkage in which, okay, the parental combination, okay, along with the parental combinations of the offsprings, okay, see, when new combinations occur along with the parental combination, then it is called incomplete linkage. Okay, incomplete linkage means we can find parental combination, but we can also find new combination. That means recombination is possible here. Okay, if recombination is possible, then it is called incomplete linkage. When the recombination possible, see here, I will draw the diagram. If you consider as a chromosome, okay, homologous chromosome, then If the genes are located very close to each other, then it is called complete linkage. If they are located bit far from each other like this, then what will happen? If you consider this A and B, after crossing over, what will happen? One of the chromosome will get A here and other chromosome will get B on its strand. Yes or no? Here B. So this is called as what? Incomplete. But always this cannot take place. That is why they have mentioned new combinations also appear along with parental combination. So that type of linkage is called as incomplete linkage. Incomplete linkage. And see here, the new combinations are formed due to crossing over. New combinations. Of course, the new combinations will be formed due to what? Crossing over. Here see, the parental combination is A, B, and here nothing. But here in this one, the parental combination is A and B. Yes or no? A 0 and B 0 there. That is why new combinations are formed. Is that clear? And new combinations will form due to what? The phenomenon of crossing over. Next one, the percentage of new combination is equal to the percentage of crossing over. Is equal to the percentage of what? Crossing over, or is less than 50%. Next, distance of the genes or distance between these genes can be identified by incomplete linkage. Next one, see, the unit is centimorgan. It is called as centimorgan. The unit to measure the distance between any two genes is called as centimorgan, because Morgan have proposed the linkage, and he has proposed what? The linkage in Drosophila. He has studied the sex linkage in the Drosophila, right? Next one, factors affecting crossing over. Factors affecting what? Crossing over. First one, distance. Distance will be affecting the crossing over as well as linkage. If the distance is more, crossing over will be more. And if the temperature is more, see, this indicates more or higher. If the distance is more, crossing over will be more. If the temperature is more, then crossing over will be more. X-rays, if it is given in higher level, then crossing over will be again higher, right? Age, if it is less, age if it is more, age if it is more, crossing over will be less. Then sex, in case of males, crossing over will be less. In case of males, crossing over will be less, as you have already studied about what? The less crossing over can be seen in the Drosophila, right? Further about cis arrangement or else the arrangement of linked genes we shall study in our next video. Thank you.